Howdy.
Howdy.
It's my honor to introduce today's seminar speakers. Today we have Dr. Heath Mills, Dr. Laura Fackrell, giving a presentation on advances in space agritech through the Rhodium Scientific Spark Program and established pathway for rapid space and R&D missions. So I'll give a quick introduction for both of them. Dr. Heath Mills is an extreme environment microbial ecologist with academic degrees from Duke University in the Georgia Institute of Technology and has held 2 faculty positions at Texas A&M University and the University of Houston. As the current Chief Scientific Officer for Rhodium Scientific, he leads initiatives to expand terrestrial and space research capacities into the advanced commercial biotechnology sector. Dr. Mills has been principal investigator and co-investigator on over 20 international space station science and engineering missions. Recently, Mills is on the advisory board for GenSpace Scholl Institute and was appointed to the ISS National Laboratory User Advisory Committee as the chair of the Technology Development Subcommittee. And Dr. Laura Fackrell is the Astrobotany and Agritech Department Lead at Rhodium Scientific. Dr. Fackrell is a geochemist and geomicrobiologist specializing in space biology and astrobiology applications. Her current research focuses on advanced sustainable agriculture and related biotechnologies for both Earth and space. Dr. Fackrell has more than 7 years of research experience in space agriculture and has led multiple publications in regolith stimulant development and best practices for regolith-based agriculture research. She has designed several regolith simulants for agricultural research. As a postdoctoral fellow at NASA's Jet Propulsion Lab, Dr. Fackrell led publications on designing applications for agriculture, waste processing, recovery, and analyzing related microbiomes for closed off-world systems. So please give us a warm welcome to our seminar speakers today.
All right. Thank you, everyone. And being up on campus, I won't have to start off this way. Howdy. Thank you very much. All right. Glad to be back up on campus. It was just about 6 short weeks ago, that we came up and gave a presentation, a very similar presentation to soil and crop science. With that presentation, we were talking about what hopefully a lot of the faculty have now seen, which is a flyer, a 1-sheet on the Spark program. That program is going to be the impetus for and the main focal point for the presentation today. Because unlike a lot of presentations I've given in the past, seminars, lectures, and everything else, this one is really designed as not so much to tell you what we've done, but more to what you can do. This is really aimed at you. So AgriLife and through the efforts of Dr. Lamb really is focused on getting more ag science into space. Engineers are great. Thank you. We have a mechanical engineering intern that Rhodium had a couple years ago. He is now, I'm going to make you do it. He's at Texas Tech. Give me some hisses. Come on. Thank you. He is a sophomore at Texas Tech, mechanical engineering. He was our intern a couple years ago. But what I want you to see is this. From the engineering side, is great to go to space, but what Dr. Lamb wants the AgriLife and Ag School and Ag departments to do is let's get biology in space and let's utilize the space environment for biological gains. And so and as I appreciate the introduction, my background is molecular genetics, extreme environment microbiology. I've looked at a lot of environments around the planet, under the planet, and now above the planet. But we focus everything with Rhodium from a science-first perspective. We are truly a biotech company. We are the first biotech space company. We were founded in 2014 by Olivia Gomez-Holzhaus. She founded the company in San Antonio. She is a Texas State grad. She's a UTSA grad. Come on. But all inside Texas, she saw a need from the industry to not just produce something out of a lab, but to have a product from a lab. Do the scientific process, not just to, you did something, but get something out of it. And so her company worked with different laboratories, universities, commercial laboratories, to improve the overall workflow, optimize those results, standardize the processes, get something more efficiently done that produces a better product. She got a contract over at JSC in about 2015, 2016, that got her going into the JSC area, got her too close to NASA, got her talking to some NASA people. She befriended one of the former directors of Johnson Space Center, George Abbey, and George said, and he is, if you look his name up, he's the most influential person in U.S. space history. He got in the program in 61. He's the astronaut maker. Every astronaut from 78 to 2003, he hand selected them. He told her, he said, what you're doing in a terrestrial lab would be great to be done on my space station. And he can say that because he's the one that actually got the space station originally funded. And he led the development. It was his station. Olivia was like, okay, that's great. You know, science and space, it's not really for me. And he told her, well, you're not a real scientist unless you're working in space. And that insulted her. And when you insulted her, she then said, I'll show you I can do stuff in space. And so she started learning why space. Why should a biologist, her background's microbiology, why should biologists look at the space environment for R&D? And so she started exploring this, and she started understanding that a lot of these processes that we do terrestrially should be applied to the space environment. And it should be what you do in your lab goes to the space environment as opposed to what you do in the lab conforms to the space environment. And so one of the big things that she did was the middle point there, the space biotech at the speed of business. The problem 10 years ago with doing anything, especially in the academic side, with doing something in space is it took about 5 years to get there. And so if you're a junior faculty, you're advised don't do something that won't give you results at your third year review and especially your tenure package. You need results. Grad students couldn't get a mission in time. You were working on somebody else's mission or you never got to see the results. So we streamlined that approach to get to space in a year, in 6 months, in 4 months. And so that's what I want to say now is that as we go through this, when we're talking about doing missions, when we high five, we've got funding, AgriLife is funding your mission, we're going to space, get ready, buckle up, because we're going quick. And you'll be in orbit in 4, 6, 7 months. That changes the game. So let's get into it. A little bit of the introduction, but let's go to space. This is the fun part. So when you think about doing research in space, with us, you'll get to go see these rocket launches. You'll get to go to Kennedy and watch the rocket launches. But your science, unfortunately, if they would let us go, my butt would be the first one to stow away on one of the vehicles and go. And if I'm missing one day, that's where I am. But our destination is the International Space Station. So when we're talking about projects today, when we're talking about going to space, this is where we go. We look at this International Space Station as a field station. When I was here on campus, I was in the oceanography department. I did biological oceanography over there, and I made a lot of students real upset because I was not doing whales, dolphins, or sharks. I was doing microbes, I was doing geochemistry, everything they didn't want to learn about it. That's what I was doing in biological oceanography, but I worked on a lot of ships. I went across the Atlantic, I worked in the middle of the Pacific. I did a lot of field station work. This is our field station. You send research to here. You take advantage of the environment that is here. Your incubator in your lab is a great incubator. It should be very well calibrated. It should be well utilized. The only difference between the incubator in your lab and the one up here is this one's traveling 17,500 miles an hour. It's still an incubator, but it's in a unique environment. And that's the environment that we want to understand how it can impact your science to show you something completely new, something that you cannot do back on Earth, you can do here. And it can only be done in this environment. Now, fun part of this also is now Kate up in the corner is actually now, segue, part of our team. And so we worked with Kate when she was on the space station. We were actually hanging out with Kate last night down at Kennedy or down at Johnson Space Center. Our team are scientists. We do have some engineers. Thank you. But we are a bunch of scientists that look at these missions, look at these opportunities. And we met with a handful in the room here today and hopefully talk to more people later about translating that why, Why should your science go to this environment? We'll develop the objectives, the downstream analytics, make sure the sample numbers are right. This is where come up with the questions. Tell us your research. Tell us what you want to do. And let's find a way to get it into the space environment and get ridiculously new and novel products out of it. Now, going to space means you are going to do a lot of pre-flight prep, pre-flight optimization. The Department of Defense, Department of War, sorry, DOW now, challenged us about 5-6 years ago to fix that speed problem. They told us, hey, if we have something that is of national interest that we want to test it now, not in 5 years. If there's a vaccine that needs to be developed, if there's something, a countermeasure that needs to be developed, we need to have mission readiness. So can you start a mission, launch the mission, return the mission, and give me a report in less than 12 months? We did it. And they said, you got lucky. Do it again. We said, give us the funding. They said, yes, do it again. We did it again. And we did it 5 times in a row. And they were like, okay, we got it. You can do it. Good. Because what came out of that was the quick space process, the quality industry compatible space process. If you work with NASA, at some point, you have to have an acronym for everything. So good luck on that one. But it was quick. But the first thing had to be was the quality, because you have to trust the results that come out of it. When you are in your lab, if I don't see some head nods from some faculty, I'm going to know you're asleep. You have to take the notes. You have to use your lab notebook. You have to record what you do. Whether you did it right or did it wrong, record it. Because some of the greatest experiments, some of the greatest results came out of mistakes. That's the reason my lab was so successful here, because they just constantly made mistakes. And they came up with new ways of making mistakes. And I was like, quit challenging this. So, but do it with the quality, the replicates, the controls, the note-taking, the standard operating procedures. Make sure you can replicate it, quality and fast. And so we got the work through the NASA processes, work through the science processes to develop that process that then has led to us doing a lot of really fun missions. So we've gotten over 35 missions, either flown. I think we've actually flown 31. We are in prep with about 6 right now that are coming up, launching in the summer and the early fall. So over 35 missions have followed this process now to do everything from stem cells, and I'll talk about a lot of different ones, but a lot of different types of science follow this same standardized protocol. When AgriLife selects your project, your research, we will roll through this same process. It is now choreographed. It is now well understood. You will throw curveballs at me. I couldn't hit them in college, but I can hit them now. And you can get through these steps to get to that launch sequence in a really short amount of time. Now, this is our obligatory hardware slide. If you do anything with space, you have to at least throw some hardware on the board. This is where this discussion is going to end, and it's going to focus all on the science. Because as you are developing your experiment, what you want to send to space, I want you to forget this, okay? Do not try to fit your round peg into one of these square holes. That's our job, okay? We will do the translation. Don't constrain, oh wait, he said he could do 36 samples. No. If you need 48, talk to us. If you need 10 milliliters, and I said this holds 5, tell me 10 milliliters, okay? Let's work on trying to have it be as much like what you do on your bench as possible rather than fitting into these. Now, the fun part with these is the left side, the Rhodium Science Chamber facility, is a facility that our founder, Olivia Gomez Wholesales, won the 2023 JSC Director Innovation Award for this facility. And it made a lot of the engineering companies really mad, which we loved, because this facility is basically test tubes, petri dishes, and culture bags flying in space. It is not an over-engineered widget that has lots of bells and whistles and can barely do any science. This is where if you need 5 milliliters, we can do 5 milliliters. If you need 50, we can do 50. If you need 500, we can do 500. If you need 10 samples, 100 samples, we've flown 750 samples. If you need photosynthesis, if you need gas exchange, if you need anaerobic, if you need subsampling, if it needs to be frozen, basically this configuration allows us to say, yes, we can do that. It's not confined to a rigid structure, which allows us now to do a lot more science, which means don't look at the hardware, just tell me the number of samples and what you need to do with them, and we'll fly that config. Now, we also have some powered hardware over here that gives us the chance to fill some gaps. There are some hardware on station, there's some facilities on station, but this allows us to fill some scientific gaps. The one in from the right and top one, the box thing up there, that's our centrifuge. So, and this is where I get to point to the intern. So in his first couple days as an intern, we blew his mathematical mind and made him do more math in a couple days than you had to do in an entire year. What this does is this allows us, it's a low-speed centrifuge, so we're able to spin samples at fractional gravity. So yes, on station, it's microgravity, roughly zero, unless you're a physicist. Microgravity. You spin this at the right RPMs, which he had to calculate. He was right. We double-checked his math. You spin it at the right RPMs, you can get lunar gravity. Spin a little faster, you get Martian gravity. Spin a little faster, you get Earth gravity. So now you can test the continuum. What happens at the fractional gravity? We need to get prepared to go to the moon because lunar gravity is obviously different than Earth gravity, it's different than microgravity. And through our biomanufacturing program we did with DARPA, we showed that microbes actually, the engineered microbes that we tested, the synthetic biology organisms, produced higher production rates, had higher production rates at lunar and Martian gravity than they did at 0 and 1. So figure that one out evolutionarily. They were better adapted to lunar and Martian gravity than they were to Earth and 0. So we have to reset all of our metrics when we go to the moon. So here's a chance to start testing lunar regolith samples, plant growth in lunar regolith and lunar gravity cycles. Yay. So plant chamber in the top right corner, that chamber up there has flown 2 missions. Total of the last mission, we flew 24 tomato plants. Light cycle, we've actually adapted that configuration to now fly 48 small plants, small cuvettes, early stage germination, so that we can grow plants on orbit, we can do fluid exchange, we can do whatever you need to do, but that's the engineering side, and I'm not that guy. So let's get to the why biology, okay? This is now where I want you to start thinking about your research. And students in the crowd, pay attention to your research in this. And when we get to the end, when we start talking about applying to the SPARK program, make sure your advisors apply your research to the SPARK program, because then you're the ones that get to go to Kennedy to go see the launch. So be selfish. So in space, we talk about the environment. The environment does a couple things to biology. So first off, you're in a low-dose radiation environment. You're in a unique, stressed environment. In a stressful environment, biology changes. Evolution 101. You have to be stressed out to change. Otherwise, you sit on the couch and watch basketball games. So you now start to see genomic shifts, permanent mutations, mutational shifts because of the space environment. These can be advantageous. This can be deleterious. This can be novel changes that may impart new functionality to those organisms. That's a permanent genotypic change. A phenotypic change, you now start talking about temporary shifts, a space phenotype. It's like if we travel anywhere outside of Texas between October and April, we freeze to death. We learn to figure out where a coat is. We put it on temporarily. We come back to Texas. We warm up. We changed for a little while, but we reverted back to the Texas phenotype. Well, in space, you see organisms that grow faster, that grow slower, that produce different secondary metabolites. That may be novel, that may be useful, but it changes when it comes back to Earth. So capture the transcriptome, capture that shift that is seen in orbit, engineer it back on Earth, and now you have a new organism, a new trait, and you've re-engineered that space phenotype for a terrestrial model. You can do that. Structural changes. And structural changes in this, we usually talk about 3D cell cultures, that side. From a plant perspective, think about how a plant grows in a space environment. Okay. You don't need a lot of structural mechanical waste of carbon there. Cellulose, lignin. Why? It's not going to fall over. And so plants start growing much thinner stems. The branching structures are much thinner. Leaves get bigger. Fruit can get bigger. Your carbon utilization is no longer going to structure, it's going to function. Okay, it's a change. You can look at different ways that plants grow. That means you can take advantage of the space environment to produce a completely novel structure. That's fun. That's what we like to do. So the other areas, and I'm going to switch it over here to Laura in a couple seconds. The other areas of research that we're doing that you may have friends in other departments around campus, or you may want to collaborate and be more interdisciplinary. One of the big areas that we work on is Regen Med. Now, AgriLife has a human health component to it, has the nutritional science side. And so this applies to those departments as well. And Dr. Lamb said that is of interest to him. Hint, hint, whenever a program manager or someone selecting a proposal says it's interesting to them, go in that area. Astronauts, as soon as they get to space, start to have age-related maladies. You fly a 35-year-old, very fit, military-trained, top physical specimen to space, and they get pre-Alzheimer's, they get pre-cancer, pre-diabetes, osteoporosis, cardiovascular issues, ocular changes. They start seeing signs of an aging population, which means in that space environment, you can detect the triggers that start turning us into older citizens. When they come home, a lot of those revert back to being 35 years old again, which means the body can turn off Alzheimer's, can turn off osteoporosis, can turn off diabetes, can turn off cancer. But I guess we lose that capacity as we get older. What if we figured out what those triggers were? That's a heck of a thing to find in the space environment. As we start seeing these changes in the Regen Med side, we've worked with teams doing stem cell projects. Stem cells change. They replicate faster. They differentiate, proliferate faster in a space environment than they do in terrestrial cultures. We had an amazing study at UPR Maya Wez out of Puerto Rico. And you work with the Puerto Rican teams. You have to have a Puerto Rican flag around you at all times, and it has to be in all the images. They're the most fun team to work with. They were awesome. Their research has now, and they talk about the speed. We awarded them the proposal, their project in March. They launched in November. They returned in December. They got a follow-on NSF grant by the next March. They published the first paper in the summer. We got our first patent with them over the summer. We've now published the second paper, just got accepted to Nature, which we think is awesome fun. This was the first paper that came out. This was published back in December this past year. The second paper is coming out now. We got great reviews off of Nature on this. But getting and seeing how we were able to utilize space environment to see very novel phenotypes for their stem cells, we're now translating that into a terrestrial model. So that was a fun Regen Med. But as we learn things from the regenerative medicine side, we go into the pharmaceutical developments. This is where nutraceuticals, pharmaceuticals, human health, nutrient acquisition from the human side. This is the food science division. We start looking at ways, new and novel food and new and novel pharmaceuticals. This goes into our bioprospecting missions that we've done, where we're looking at these new and novel compounds that are only seen within the space environment. We also worked with and just published, this paper came out in January, and the press on this one has been ridiculously fun. We did a phage mission. Phage are viruses that only attack bacteria. This means they are a novel antimicrobial, antibiotic. They don't get the antibiotic resistance that when you take penicillin or you cut your hand and have to take antibiotics. This is stupid. But it's a way of producing a new antibiotic. We published this. This has now been picked up by many different news agencies. Fox News picked it up because what we saw was that viruses became more pathogenic. They were more lethal to their host in the space environment. Okay, this is a phage, this is bacteria. Some of you I know are working on plant pathogens, and you're looking at plant pathogen interactions and the stressors they cause to plants. What happens here? And can you look at new countermeasures against plant pathogens in the space environment? Can you understand how plant pathogens work in this ramped up high evolution, high metabolic rate environment to translate that back to new therapeutics, new countermeasures for plant pathogens? Write it up, send it in. Let's see if AgriLife will fund it. The last one before I turn it over to Laura is that now we talk about production. And biomanufacturing, whether that is from algal species, whether it's plant species, whether it's microbes, bacteria, fungi, the space environment provides a very unique model for growing things. But now the important part of this is when we start talking about LEO, low Earth orbit, there's not a lot of supply vehicles that go up. There's 5 or 6 a year that go up to LEO. When we start talking about Artemis and going to the moon, you have a much bigger supply chain issue. When we go to Mars, it's worse. So understanding how we can make something in that environment as opposed to take it to the environment becomes very important. How many colonists were successful coming to the New World that had to bring all their supplies with them? Didn't last too well. They learned how to live off of the environment. So whether it is Mark Watney and on Mars and science and the hell out of stuff over there with potatoes, or it's the moon, or it's algae, or it's plants that provide dual use that's not just producing a fruit, that's producing a pharmaceutical, we're going to be doing a lot of biomanufacturing moving into the lunar system. We did this huge program with DARPA, and this is the one that Jordan was attached to here, producing the centrifuge, where over 3 missions here, there was actually a 4th mission that just came back last year to finish up the program, 4 flights in about 20 months, producing over 1,000 total samples that went to space. We really started to understand how to re-engineer organisms that responded to that space environment, increased production rates. We got overall an 81% increase in production on orbit compared to the terrestrial counterparts. So we were able to really ramp up production on this. Those translated back to engineered organisms back here, and we increased production rates back on Earth. So think about that with your plant system, because I'm going to blow your mind with that at the tail end of this. So that's 3 of our main areas we work on. Laura is our lead for the ag tech side, and so she gets to jump through those slides.
Hello, everybody. As we kind of introduced a little bit before, I'm Dr. Laura Fackrell, and so I just joined Rhodium Scientific about 1.5 years ago. And I came from JPL, where I was working on space agriculture and regolith-based agriculture and lunar simulants. I worked on that for Mars in my dissertation. Spent a little time in between there doing some astrobiology as well, so there's a lot of hats that you can wear. But I have a lot of background in how we understand how to do space agriculture. In doing that, we've also looked at how does that translate back to Earth? And so one of the things that we're looking at in Rhodium Scientific is that we want to understand not only how do we grow plants in space for space exploration, but actually how do we use the space environment to improve how we do agriculture on Earth? So that's one of our main drives. It goes both directions. It's synergistic. And so bioregenerative life support and industry production include space-grown crops and that kind of thing. But and again, that's only part of that story. A good example is LED lights. And so LED lights are originally designed for space use, but now have become like a really common product in that. So that's a technology development that helps translate out of Earth. There's also lots of other kind of things. So I'll kind of talk about some of those in some of our missions that we've gone through. But that's what we're really focused on, and that's kind of what we drive from. And a lot of those things come in from, so the technology, advanced growing techniques, the resource use and how we use resources more efficiently, and all sorts of things like that. So I'll start with a fun mission one of the early missions that I came in on was our USAFA night mission. And that's where we actually were able to germinate some small space tomatoes. So SPACE was an acronym that stood for Small Plants for Agriculture in Confined Environments. So these are plants that were designed for a controlled environment to be able to grow in a very small space. And so they're tiny little tomatoes. They can't, like the smallest version they've grown can grow in like a quarter size. And so they can grow these tomatoes. And they're actually also designed, so they did some genetic engineering to design them to use less light and to be more efficient photosynthetically. And so they're grown on 3 hours of light a day, and kind of used an alternate carbon source. They used acetate as an alternate carbon source, to be able to make up for some of that energy as well. And so that's looking at alternative ways to provide energy to the plant so that you can understand what's the balance of energy and resources and how do we produce the same yield but cost less energetically and become more economic. And so there's a lot of applications that you can do with that because that's really important for space exploration. You have very limited resources, and power is a hugely limited resource. Space is a limited resource. And so those are things that you want to maximize and prioritize in space, but those also translate back to Earth and how you do controlled environmental agriculture when you're trying to understand how you grow something in a closed room. That's also hugely important. And so we're able to grow 24 samples in that one mission, and they grew for about 30 days in a 5 milliliter tubes, and then came back and they were able to, and they actually were able to bring them back. They brought back ambient, and so they actually transplanted them, and they continued to grow them even after they returned back to Earth. So there's some pretty cool stuff that came from that. So
Yeah, Christmas.
yeah, so that was pretty fun. And so that was a really good mission to look at that. That mission also looked at we have the modularity and like the replication that we can do. And so one of the challenges of the space environment is the limitation of space. And so a lot of the traditional plant chambers on the space station do like 6, maybe 12 plants as your maximum plants that you can grow at once. But we were growing 24, so we're already doubling things. And then for our next mission, which is our microgreens missions that we do with Swinburne, we actually grew over 200 germinated. I forget. It was like over 200 seeds germinated. And so we have tons of sample replication and large replication and like that statistical robustness that allows us to do a lot of really good science. And so that's kind of one of the ways that because we focus on that science first and we're not over engineering the plant chambers, we can do a lot more replication, especially the smaller the size, the higher the replication number. So there's a lot of variability and a lot of robustness and replication and iterations that can come from that allow us to really drive the science forward and not just worry about the bells and whistles. And this is part of the Swinburne Innovation Challenge. And so we've done a lot of things with Swinburne. This is actually, they're up to their 4th mission now is upcoming with a fifth. Oh, this is the 5th mission, microgreens too. Okay. Yeah, so there's 2 microgreens missions where they both grew. So they study the germination rates on the first mission. And now on this upcoming mission, they're going to look at later stages of growth because they're going to grow them a little bit longer. And kind of follow up and understand how the microgreens respond to space. They've also done yogurt and fungus and other things too. So it's not just plant production, it's other kinds of food production as well, really important for the space environment. And this is a really good example of workforce development because they are student design projects. And so you can, as a student can take part in these things and design products that go to space and do really cool science. Another STEM workforce development kind of focus that we have is what's called the Tomato Sphere Project. And so the Tomato Sphere Project has been going on for a long time. They've flown a lot of missions, but this is now we're have come in kind of as their implementation partner. And so that means we've partnered with them and we are the ones that help get them into that space environment. And we're adding a few like tracking additional data that wasn't being tracked before. And so they send millions of seeds. They send very large pillows of tomato seeds up to the space environment. They stay there for for whatever the duration is of that particular profile, bring them back, and then those are distributed out to students who then do growth comparisons of earthbound seeds and space-flown seeds. And so that's for that STEM purpose where they can then explore and understand how seeds change when they've responded to the space environment. So that's a really cool opportunity that is able to connect to various STEM projects for grade schools, but that also connects to really powerful science that when you fly seeds in space, they do change and they grow differently when they come back. And so it's a really interesting way of introducing novel variation in that genetic profile and seeing how you can then breed new species of plants and where there's a lack and we can reintroduce some genetic diversity. And so part of this was a Rhodium Space Ag Veterans mission, where we were able to partner with various industry companies to select specific seed types that were relevant to what they focused on. And we flew over 15 types of seed types to the ISS, and we flew multiple packets of each kind of seed type. So there's lots of different samples of those packets. And they were able to go to the ISS. They were originally going to go for 4 months, but it actually extended to much longer. So it's almost about 6 months where they're up in space and they're able to understand and look at those different seed types. And so now they've just recently came back like a couple weeks ago. And so now they're ready to test and see how they respond compared to their Earth-bound counterparts. Yes. And then the last part is with my background in regolith stimulant design and regolith stimulant applications for agriculture, I've also been working a lot with some regolith stimulant thus and the proper selection of appropriate stimulants to use for agriculture, because not all stimulants are designed to use for biology or even specifically for growing plants. They don't always, the stimulants are designed for a specific purpose, and that design doesn't always take into account the needs for biology, how you would sterilize something, whether or not there's an ingredient in there that is used to mimic a certain thing, but it doesn't actually reflect the chemical profile needed. And so there's a lot of work that goes into creating a stimulant that's appropriate for biology and then how you actually handle that stimulant afterwards. And so we also have a lot of work on really creating those best practices and standardizations that allow us to do good science and to have results that are actually applicable to what you want to do. So there's also a really important work that we've been doing with stimulants in that area. And that continues from what I was doing at JPL. And I think that goes back to and so there's another, and so we're going to jump back to Heath to talk about the biobank, where a lot of these things will happen.
Okay, so I hope now you've seen a little bit about why we do what we do in space. You've seen the hardware slide. You've seen why biology changes in space and a myriad of different subjects and areas where space changes biology. Okay, now this goes as a big point back to you. Now, this is where I want you to get creative, and if I have to call on you, I will. I know one name, and I'll point to everybody else. And so I want you now to start thinking of your project, what you want to do in space, and I'm going to give you 2 ways of doing that. Okay, first off is the biobank. So we were funded through NSF, through an SBIR a couple years ago. We're now moving this into phase 2. What this does is that we've flown a lot of samples. We have a lot more coming up in a couple different launches over the summer. 1,500-ish total samples that have flown. These are seeds. These are microbial samples, bacteria, fungi. There's some stem cells, some other human cell lines. These are all flown and have been up there for different durations. If you want something that's been up there for a few days or a few months, let's talk about it, because what you can do is access some of these samples, which will give you the chance to get some preliminary data immediately, where you can change that preliminary data into a new proposal, a new area of research. You can get a publication out of this. You can move and go straight to, and what we call is you're jumping to the analytics. So you've bypassed the pre-flight, you've gone right to analytics. Now, the other thing you can do is you can grab some of these space-flown samples that have already been optimized. You have data sets from previous publications that you can stand on. You can use those. You can use these to plan for your mission coming up. So you've seen what they've done in space before. Now you're going to use these and maybe make an additive, stress them out in a new way, longer growth, shorter growth, whatever. You can now build your mission off of something that's already been flown. So these are fair game for you to use. Let us know what you have, what you need. We can maybe have something close to it. We have the exact same thing, or we may be able to put it on a ride coming up that's real quick and you get some samples much sooner. So the biobank is one that we want you to now utilize to facilitate your research and your questions. The other one is through the Spark program. And I am going to reverse these real quick, and I think I'll change that later. I always change your presentations in the middle. It's more fun that way, and it means you don't know what you're going to say next. So this is what we've now been talking with AgriLife, and Dr. Lamb has been amazing in supporting this. For the last few weeks, we've been developing the Spark program for Aggieland. The Spark program is what we were doing with Swinburne that Laura was just talking about. We've done this down in Puerto Rico. That's where the Puerto Rican missions came out of. We have these going in different areas, but now we're bringing it to Aggieland and doing it here. What we are doing, this QR code is live. The QR code is real. The QR code leads to a questionnaire that allows us to gather some information about your research ideas, what your research is, what you're wanting to do. Unlike other proposal submissions, we're not going to look at this and go, horrible, out. That's not good, and gone. We want you to have a good proposal. So submit your ideas. This is also like voting in Chicago. Vote early and vote often. So if you have 4 ideas, submit them. This is not limited to one. If you want to submit with another faculty member, please do. If you want to submit with another faculty member in another department, awesome. Submit. Students, get your advisors to submit, because if they submit your project, what you're doing, you're the one that gets to go to launch. Submit. What we're wanting on this is, what are your ideas? My research is on root growth and how roots affect stress within the root system overall, affect the plant growth and the fruit production. How is that applicable to space? I'm not sure. I'm going to submit my research and let's go talk to the Rhodium team. What we're going to do is we're going to take all of the submissions on these, we're going to go through them, and we're going to most likely ask you some questions. You said you need 26 samples. Why 26? Why not 36? Why not 16? Do you really need to fly coconuts? Don't do that one. Don't do that one. So what we want in the end and what we're going to turn over to AgriLife is all these submissions. Rhodium is going to then say, are they flight feasible? And so we want, if we get 30 respondents, we want all 30 to be flight feasible. So we're going to work with you to make sure that they are flight feasible. We're going to turn these over to AgriLife. We are going to assess the scientific value. So there are scientific value, there are scientific points, there are things that are being done in space right now that have higher value than others. To a huge hint, and if I offend somebody doing a Arabidopsis, sorry, NASA doesn't want to fly a Arabidopsis anymore. They want to fly something that has a dual use. You can understand phototropism, gravitropism on a food crop rather than on Arabidopsis. So they want food crops. They want something that the astronauts can eat, something that is functional to a space mission. So we'll help you and we'll evaluate the science side of this. What AgriLife is then going to do is they're going to look through the respondents and they're going to say, this is of interest to us, contact this professor and let's get them going to space. There is funding attached to this. So we will be asking you also for what your lab needs in terms of funding. In the questionnaire, there is a request for funding listed in there. The caveat on that from AgriLife is if you ask for $20,000, they're going to say, great, we will give you 10. We'll give you 15. We'll give you some amount less than 20. Can you have a little skin in the game? Can you provide a little bit of funding? That could be you will sell them a grad student. I don't blame you. I had several I wanted to sell. You could provide them a grad student time. It may be one of your summer months. It may be from your startup package. It may be from another grant. It may be from a return of indirect. Something they want to have as your buy-in to this. We're doing the same thing as the company. We are going to get some funding out of this, but we're also contributing a lot to this because we are investing in the Aggie environment and we want to see this science go. So what are they looking for? That's why I got to reverse these slides. So what are they looking for? We've been talking to a handful of people today. We talked to a handful of people the last time we were here. We have some more meetings tomorrow. Ping us, talk to us, because we'll help you with what are they looking for. So in these areas, the bottom half, plant genetics, the human health and nutrition, water resource management, phytomining, nutrient cycling, those are all areas that Dr. Lamb specifically said, I personally am interested in these. So if you have a topic that fits in one of those, make sure you submit it because the person that is selecting what goes forward said they're interested in those. Okay? That does not preclude the first couple. Any of the tropisms, phototropism, geotropism, picotropism, those are all really interesting fundamental science that can and should be done in the space environment because you cannot do that back here with isolating them. The only way to do true testing of gravitropism is to go up there and start playing with the gravity side, vectors. It's fun. Those are fundamental science, right? Now, for the AgriLife Spark program, for Texas Space Commission, how many people submitted to Texas Space Commission and is upset that they submitted to the first round? Yeah, it's horrible. It did not, they screwed it up. And I can say that Norman, if he was here, he would admit They screwed this up in terms of addressing the faculty and the academic side. The proposal and how it was written did not facilitate academic research. It didn't. They are fixing the glitch. So there is $300 million that's going to become available within the next few weeks in 5 areas. And this is where I get to try and do the 5 areas again. Can I do it? LEO, low Earth orbit. So something research that can be done in low Earth orbit, something targeting the moon. Great for this crowd. Lunar work, lunar regolith, anything on that side is being targeted. Propulsion systems, probably not this group, and I'm not doing that. And you can do that. Propulsion's not that. Human health and performance, so human activity, the nutrient people, that one definitely, and playing into the plants and providing nutrients to the crew, that one you can do. And then workforce development, that one's obvious. So definitely, these are the 5 subjects that they're going to target. Whether or not they're looking for proposals that are in the $100,000 to $1 million range or $1 million to 10 million range, how they're doing that is unsure at the moment. But when you submit to the QR code, to the questionnaire, we are going to also take these proposals and tell you to get ready for TSC. Okay? TSC and AgriLife want fundamental projects like the top one to have an applied nature. It needs a little bit of a commercial side to it. It needs a little bit of terrestrial implication. It can't, they don't necessarily want it to be space for space. If you're learning something and utilizing the space environment to translate it back to ag in Texas, you rank higher. Okay? That translation side, that dual use side, that is something we can definitely help you with. So if you have questions within the questionnaire, we will be sure to answer your question as fast as we can figure out the question, and will be less questions. So that is something we can help you out with. The other fun one that we are slowly talking about and developing and working on, and it stems from, pun intended, the UCR mission, is the modifying the diurnal cycles. We talked about this at lunch today. This is where I really challenge this environment to think outside the box, and the box is Earth. There are certain things that biology has been constrained to because biology has been on this planet. First off, gravity. Up until the last 80 years, life has never existed outside of gravity as far as we know. Mars, different story, but we'll talk about that later. Martian. But gravity has been a constant. The other constant has been a diurnal cycle of about 24 hours and about 12 on, 12 off. Depends on the time of season, I got it. But you've been stuck to a 24-hour day. We don't have that problem anymore. Last 100 years, we developed these, and we're about to go off Earth, where what's a day? We're about to go to the moon, where it's 14-hour day, 14-hour dark. That's not a day. We're about to go to Mars. It ain't 24 hours. Days doesn't matter anymore. So what if you engineered biology to no longer recognize a day? What if you sped up a plant cycle to grow twice as much in one day? What if you did a light dark cycle of 6 hours on, 6 hours off, and you got 2 days in one day? What if you did 3, like we did with UCR, and had a 3-hour dark cycle? What if you could get 4 crop cycles in 1 year? That disrupts all of an ag system. We have the genetics to do this. We have a test bid to go test this, and we have a need to produce more food in a smaller space, both on Earth and off. So what if we threw a diurnal cycle out the window? What if you changed the light cycle? What if you changed the LED lights that came from space? What if you changed the water resource management? What if you changed the nutrient cycling? Let's utilize the genetic engineering that's here. Let's utilize the plant and crop science that's here. Let's utilize the food production that's here. Let's change the model. And I think that would be huge for AgriLife. It'd be huge for A&M. It'd be huge for TSC. So I really want you to think outside the box on this, and let's go do something fun. All right? Okay. I'm done 12 minutes early. Perfect, because I want to throw this back to you guys. This is where we get to play improv. So questions. I'm going to stop here. I'm going to pause here and I want questions. I want ideas. Let's have a discussion. Awesome. Yes, sir.
Does that require citizenship?
For the AgriLife program, Spark program, no. For TSC, I don't know. For involvement in NASA and going on site on NASA to do and be involved in the pre-flight launch sequence, because we usually go about 7 days before and do everything there, depends on the country. There are ways to get pretty much anybody on, but let's talk about that because that is, I mean, you're here, you're an Aggie student, you're here. So let's talk about that. Good, great question. Great question. Other great questions. Yes, sir.
Great presentation.
Thank you, sir.
What are the seeds available already in the space and if we can use it? Do you have some tomato seeds you can test?
We have tomato seeds.
Okay.
Yes, we do. So for the seed question, and this is the fun one, and getting a little bit more into that. So there's a lot of papers out there right now that talk about seed production coming back from space, the good, the bad, and the ugly. The overarching question or result on this is the seeds change. Why is a huge question yet to be determined. There are some studies, one out of Sri Lanka with cilantro seeds, the coriander seeds, rather, same thing, coriander seeds that saw a 30% increase in leaf production just by going to space and coming home. And they didn't do anything. They just rode up and came back. Is it something with the low dose radiation changing the protein structures that affect the overall seed? Don't know. But if you want to test some seeds early, let us know. If you want to propose to fly a lot more seeds and a lot more seed varieties, yes, yes, and please. We can fly lots of seeds. So yes. And we are also, because of the Space Ag mission that Laura was talking about, we are looking for partners to go in and analyze some of those seeds that we got back, because we would love to go in and get the genome, the transcriptome, the proteome, the picanome out of those seeds, look at overall germination rates compared to the ground controls. So we are looking for partners on that project. So please. Thank you.
On the same question, on the tomato, do you have control? I'm the one which is space perfect.
Yes, from the same lot.
You can compare for the human health aspect.
Yes, sir. Yes, sir. And that's for the samples that we have in the biobank, short of a couple because of some issues with different things. We have the ground controls and the flights for all of them. Those that we don't have specific ground controls, they're lot generated, and so we can go get the same lot. The other one that I want to understand out of some of these samples and really get into the genetics of it is how variable is it? Do we see, if we fly the same tomato seeds on 3 straight missions, do we come back and get the same results, or is it all over the place? We don't know that right now. And if it's more reproducible, then it doesn't matter what the ground controls were, as long as it's the same seed type. But we don't know that answer yet. That needs to be done. And that's part of the reason that the biobank is so important, is we're trying to standardize those samples so that from mission to mission to mission, we don't have interflight variability. When we did the biomanufacturing mission, the DARPA biomanufacturing mission, because we went so fast on the first mission, we flew from kickoff to launch was about 4.5 months. We were supposed to do it in month 9. We got it real quick. Because we did that, we actually flew a mission 1B at the 8-month mark, where DARPA asked us to fly the exact same samples again, analyze those, and look for interflight variability. And we actually got, within statistical error bars, the same results. So we were able to see with a microbial population, the same results being treated the same way on 2 different flights. So that one we were able to show.
On the TSC, do you have any expected date when they're releasing the
We have nicknamed the program Coming Soon. But given hints that we're seeing from TSC, and we're in constant comms with the TSC teams, the TARSEC teams, they're saying April. And given what's just happened in the last week, it looks like April may be the real thing. Now, with that, and if they go the same path that they did the first time, they gave a real short window for proposals. a ridiculously short window. I expect them to do that again. And that's why getting everything into the questionnaire where we can now get the flight feasibility figured out, we actually got awarded one of the first proposals. So we were partnered with Intuitive Machines. We're building a free flyer vehicle. It's called Zephyr. So we are actually one of the performers from the first time. There is a little bit of a thought of a route where if a proposal can target and mention that Zephyr could be a future direction, that we may be able to get into a special queue. And so we'll talk to you about that. And with the proposals that are coming in, if we can at all list Zephyr as a future direction on this, and what that provides is scalability. On-demand flight sequences where you're not waiting for SpaceX to launch and come back and getting Butch and Sonny stuck up there forever and all that stuff. We can launch when we want to, but it also allows for larger volumes. And so if we're looking at growing 6 plants is great, but if we can grow 60, that'd be even better. We need our own vehicle. We can point to Zephyr. There may be another funding route on that we can talk about. The other thing with funding, and I will say this, let me jump in here real quick. Talked about AgriLife funding. We talked about TSC funding. The other thing that we're doing with these questionnaires and what we've been doing and being very successful with over the last 6 years doing all these missions is that we talk to a lot of program managers within DARPA, DITRA, NSF, NIH, pick some letters over there. ARPA-H now is a lot of fun. And a lot of the program managers literally say, I've always wanted to do something in space. What do you have that fits my program? And we go, we have these 10 projects right here. Great, let's do 1, 4, and 7. I want to fly those. And they'll select the projects. So from these questionnaires, when we talk to a program manager that says, hey, I'm really looking at countermeasures against plant pathogenicity, what do you have? Oh, I've got these 8 projects here. Okay, so in the past, that's led to either them opening up new programs or them directly funding something that's there. The other one is if you currently have funding, it may not be NASA related. NASA funding is NASA funding, great, congratulations, you got that, that's amazing, but it could be any one of the other agencies. There are supplemental awards that can be awarded to do extra objectives. And we talk to the program managers and say, hey, the work they've been performing for you and doing very well for the last 2 years should go to space. We have a launch coming up in 6 months. If you want to turn on a little bit extra funding to them right now, they can be in space in 6 months, and your program now can say they've done something in space. How do you like that? And they go, we love that. And so we've gotten funding. The phage mission that I talked about, that was a supplemental award to the University of Wisconsin team. They had been performing on that grant for 2 years. In their 3rd year, they went to space on a supplemental award, and they got additional funding. So anything that currently has funding, let's talk about adding an objective and doing a supplemental award. And we will work with the program manager to demystify the space process and say, no, seriously, this is real. And we'll work with you on that side. So that's another avenue. And that's why submit early, often. Current deadline, though, and if you look at the flyer, if you go on the website, current deadline is next Thursday, sorry. It is 7 days. Today's Thursday. I don't know what it is. First day of the tournament. Should be Thursday. Week from today is when the proposals to submission stops. Don't be like me and wait to the last minute, but I know most of you will because that's only way anything gets done is have a deadline. Talk to us. And please do not spend so much time to try and make it perfect. This is a work in progress on this one. We are not an NSF nasty reviewer. We are trying to get you to win. So submit. Other questions, other ideas? Who in here thinks they may have a research area that's not applicable to space? Challenge me on this one. Let's do this. Yes.
So some things maybe have seed-borne diseases like that. That's what the control measures use. So for the germination process, we won't get all 100% germination. It will be caused by disease or maybe it has any other issue. So that's what we will do.
So different germination rates, you're 100% right. And flying to the space environment, now what are the germination rates when they return home? Understanding variances in those germination rates, to me, I would want to go in and start understanding the why, what, and how, and why is there a difference in the germination rates. What we saw with the tomatoes was impressive. We actually got 23 of 24 to germinate in space, and that beat all of our ground data. That was better than our ground data. We got 23 of 24. Now, to go into that question, especially with pathogens on that side, one advantage that we have, so if anybody's ever looked into what's available on the space station to grow plants, you have the advanced plant habitat, which now if you propose to go in there, if you're lined up to go there, I hate to tell you, it has been discontinued, it has been trashed, and it's no longer available. So do not propose to APH, it's gone. Veggie was the other system. Veggie is still available, but in order to do germinations through veggie, you had to sterilize your entire system. And so, yes, that works well at getting rid of pathogens, but that's not natural. And a lot of what is on the seed is a natural microbiome that helps prevent pathogens. It helps with overall nutrient acquisition. It's good for the seed. So one thing that I would love to understand is with the spaceflight environment and coming back with a new novel germination rate, overall plant fecundity, everything with the plant is different. Is it because the plant seed changed, or is it the microbiome on the seed that changed? Because we know the bacteria change from the gut microbiome to, we did do a soil rhizosphere project, the soil rhizosphere changed. We know the microbial community on the seed's changing. So is it the microbiome that makes the seed grow better when it comes home, or is it the seed itself? Don't know. So looking at germination rates and looking at pathogenicity with return would be amazing to look at, because now you also then want to see, and can you develop any countermeasures out of what you see? We lost these 3 members of the population and the germination rate tanked. Well, they may be important. These 3 members increased in abundance and the germination rate went up. Maybe that's there. So really good, really good. Definitely throw that in and let's talk about that more. Yes, ma'am.
You tried that soil microbiome in space and the soil microbiome in the present ground media. Is there any, what is the difference, major difference? Did you check like that?
So we did. There's a project, and then the data set's going to come out of Cornell out of that one, where we flew different live microbial rhizosphere populations in different soil matrices. There was a biochar, there was a potting soil, and there was something, a vermiculite or something. It was more of an artificial, I forget what the third one was. But yes, there were variances, and before the paper comes out, I will tell you that we did see a rise and fall in different members of the population. The overall community structure did change. We did not, nothing was germinated out of this. This was just packing of live soils and live soil equivalents. So that did change. Looking at the way that the plant, the germinating seed, the microbiome changes together and those interactions together would be very interesting to look at. The tomatosphere project used a gel-rite, an inert system. What? Eusophonite. Sorry, yeah, eusophonite one. Had an inert soil matrix, a gel-rite, that we did add acetate to several of them because of the genetic modifications to the tomato seeds. But looking at those interactions, in veggie, you can't fly live soils in veggie. With our systems, you can do live soils. We have the containment to be able to do live soils on ISS. And with the centrifuge, you could do live soils, live regolith would be interesting. But you could do regolith, you could do live soils in a fractional gravity and also look at germination rates in that. Please, somebody propose that one. We both want to do that one.
Mr. PhD, is probably interested.
There it is. Good.
Not on the space science, but they can add one of those.
She's going to be space science now. So, and with this is what's been interesting. So with our DARPA program, so the lead team of the DARPA program was University of Florida. Well, I'll hiss that one. I was at Florida State. So the lead team out of University of Florida, the graduate students from there 3 of them have now graduated with their entire dissertation on space flown samples. They've gotten 3 and they've been involved in 3 missions. They've gotten multiple publications and their entire dissertation has been on their own space flight projects or multiple missions, which that is awesome that they got that much production out of it. So, challenge. Cool. Any other questions? I'm over time, and that usually happens. But any other questions, come see me afterwards. Laura, nice job. Thank you very much, ma'am. And thank you for letting me pick on the intern. Nice job, dude. All right. Thank you all very much.